morning, everybody. Today's topic for lecture is cerebellum. Next. Cerebellum, it occupies, this is the cerebellum, and it occupies the occipital fossa, posterior cranial fossa, or the occipital fossa, and it lies just above the medullary region and behind the pons region. Next. Cerebellum consists of two hemispheres. These are the two hemispheres, and they are united by a vermis. This is the vermis area which unite the two hemispheres of cerebellum. Next. The cerebellum, it consists of anterior, it consists of anterior lobe, posterior lobe, and the vermis area. The anterior lobe, in case of cerebellum, if we see the internal structure of cerebellum, we can see that the outer margin, it consists of medullary center, that is the white matter, and the outer margin of cerebellum, they are the cortical cerebellar cortex. And there are deep-seated nuclei which are there in this. One is the dentate nucleus, which is these in deep nuclei, they are embedded in the medullary center. The dentate nucleus, fastigial nucleus, globus nucleus, and emboliform nucleus. These globus nucleus and emboliform nucleus, they are together known as interpositus nucleus. Interpositus nuclei. This is dentate nucleus, fastigial nucleus, globose, and emboliform nucleus. The globose and emboliform nuclei, together they are termed as interpositus nucleus. Next. The cerebellum is divided into, from here you can see that these are the hemisphere and this is the vermis area. From here you can see the physiologically cerebellum is divided into three divisions. One is spinocerebellum, or it is also known as paleocerebellum. This area, it consists of vermis, and this is the vermis area, and the outer one, intermediate hemisphere. This whole region is known as spinocerebellum or paleocerebellum. This area is concerned with the control of voluntary movements of the limb and trunk. Now, this lateral portion of the hemisphere, it is known as cerebrocerebellum. Cerebrocerebellum or neocerebellum. The outer one, the area outer to the this intermediate hemisphere, this area, this is known as cerebrocerebellum or the neocerebellum. This function in conjunction with motor cortex and the association area, and this area is involved in planning of the movements. The third division of cerebellum, it is the vestibulocerebellum. Vestibulocerebellum or the archicerebellum. This area is responsible for the control of eye and head movements and the balance of head on the body. This area, this vestibulocerebellum or the archicerebellum, it is responsible for control of eye and head movements and the balance of the body. So there are three divisions of cerebellum. One is spinocerebellum, also known as paleocerebellum, cerebrocerebellum, also known as neocerebellum and vestibulocerebellum or the archicerebellum. So the three divisions of cerebellum, physiologically, these are the functional divisions of cerebellum. Functional divisions of cerebellum. If we go for the internal structure of the cerebellum, the cerebellar cortex, it has got three layers, and the three layers are molecular layer, Purkinje cell layer, and the granular layer. Then the corpus, that is the medullary center, and in the medullary center, as you have seen in the diagram, there are four nuclei, that is vestigial nuclei, then the nucleus interpositus, which is divided into embelliform nucleus, globose nucleus, and the dentate nucleus. This is the internal architecture of cerebellar cortex. The cerebellar cortex, internally, it has got three layers. Three layers, as you have seen, that is the outermost is the molecular layer, then Purkinje cell layer, and the granular cell layer. Before discussing the internal structure of the cerebellar cortex, there are three peduncles through which the input and the output from the cerebellar, it takes place through these cerebellar peduncles. One is inferior, superior, and the middle peduncle. Most of the incoming fibers, they come through the middle and the inferior cerebellar peduncle, and most of the output, majority of the output fibers, they leave the cerebellum through superior cerebellar peduncle. The fibers which are the tracts, the cerebellum, it receives the impulses from almost all parts of the body. It is from the extremities, from the head region, face, head region, and the brain also. So the cerebellum, it is sometimes also known as the small brain of the body. It is also known as the small brain, as it receives the signals from all over the body. The sensory tracts which convey the impulses from the extremities of the body, they are the dorsal spinocerebellar tract, ventral spinocerebellar tract, then cuneo-cerebellar, rostral cerebellar tract. Now, this dorsal spinocerebellar tract, it conveys the impulses, it carries the impulses from the upper and the lower extremities, and it enters the cerebellum through the inferior cerebellar peduncle. The anterior and spinocerebellar tract, or the ventral spinocerebellar tract, it doesn't send any impulses from the extremities of the body. Rather, it sends the impulses from the anterior horn of the spinal cord segment. It sends the efference copy, that is known as it sends the impulses which are coming from the descending, which are conveyed to the anterior horn through the descending tract, and the impulses which are received at the anterior horn, the ventral spinocerebellar tract conveys those impulses to the cerebellum via superior peduncle, and that is known as, we can say that ventral spinocerebellar tract, it sends the efference copy to the cerebellum. It sends the efference copy to the cerebellum. Now, the cuneo and the rostral spinocerebellar tract convey the impulses from the head and the neck region to the spinocerebellar tract. The other areas from which the spinocerebellar, input which cerebellum it receives input from the cerebral cortex via pons. And then it also receives the input from the red nucleus, then vestibular nuclei, then the brain reticular formation area, from all the area, brainstem. Then it also receives information from the basal ganglia. Then it receives cerebral cortex via pons, then red nucleus, reticular formation, and from the lower part of the body through spinocerebellar tracts. So cerebellum, it receives, as you can see from here, that it receives the information from all parts of the body. And that is why we say the cerebellum is also, that is why the cerebellum is also known as the small brain. Now, the internal cerebellar cortical structure, once we understand this internal structure of the cerebellum, then only the functioning of cerebellum you will be able to understand. It is three-layered structure. Outermost is the molecular layer, then Purkinje cell layer, and the granular cell layer. Now, the input which is coming to the cerebellum, it comes through two fibers. One is Mosses fiber, another is climbing fiber. These are the two fibers which bring the input to the cerebellum. These climbing fibers, they are the fibers which arise from inferior olivary nucleus. Inferior olivary nucleus. The climbing fibers are coming from inferior olivary nucleus. The spino-olivary tract convey the information from the various parts of the body to the inferior olivary nucleus, and from the inferior olivary nucleus, the climbing fiber, they bring the impulses to the Purkinje fiber. They synapse with these climbing fibers. One climbing fiber synapse with this climbing fiber, it synapse with the Purkinje cell. Purkinje dendrites as well as the cell body of the Purkinje cells. Now, whereas the Mossy fibers, the rest of the tracts, the Mossy fibers, they are the second order neurons, and most of the tracts, the rest, except for olivocerebellar tract, all rest of the tracts, incoming tracts, they are made up of Mosses fibers. These Mosses fibers, now the difference between these climbing fibers and the Mosses fibers are, the action potential generated in the climbing fibers, it is a complex spike which is generated, whereas in case of Mosses fiber, it is the simple spike which is generated in the Mosses fiber. The weak action potentials, they are generated in the Mosses fiber, whereas in the climbing fibers, strong action potentials are generated. They are also known as the complex spikes. And the weak action potentials which are generated in Mosses fibers, they are known as the simple spikes. Now, these are the afferents. From here you can see these climbing fibers and the Mosses fibers, they form the afferent or the input, which are, they are the input carrying fibers. Now, these, the climbing fibers, the Mosses fibers, let us first discuss about some Mosses fibers, how they work. The Mosses fibers, they excite the cells in the granular cell layer, the granular cells. Granule cells, they are excited by the Mosses fibers. This granular cell layer, it contains the two types of cells are present in the granular cell layer. One is granule cells, another are Golgi cells. The granule cells, their output is excitatory. They are excitatory to Purkinje cells and the other types of other cells which are present in the upper layers. The Golgi cells, they are the inhibitory interneurons. The Golgi cells are the inhibitory interneurons, whereas the granule cells, their output is excitatory in nature. How these Golgi cells, they function? Whenever these Golgi cells, they are also excited by they receive the impulse from the Mosses fibers, and these Golgi cells, they are responsible for, once they are excited, they inhibit the granule cells, or they control the output of the granule cells, and this is known as feedback inhibition. This is known as feedback inhibition. For the feedback inhibition, these Golgi cells are responsible. And why we say that these are feedback inhibition? Because once these granule cells, they receive the impulse via Mosses fiber. These granule cells, they send impulse to the Golgi cells also. These Golgi cells, in return, they inhibit the granule cells, and they control the discharge of impulse from the granule cells. And that is why they are responsible for these Golgi cells, or the inhibitory interneurons. They are responsible for feedback inhibition of granule cells. Now, the other type of interneurons, which are also, there are two more types of inhibitory interneurons which are present in the molecular cell layer, and they are the stellate cells and the basket cells. Basket cells and the stellate cells which are present in the molecular cell layer. These are also inhibitory interneurons. They are inhibitory in nature, and they are interneurons. How do they, now, these and the Purkinje cells, once these granule cells, when they are they receive the impulse via Mosses fibers, they excite Golgi cells, basket cells, and the stellate cells, as well as the Purkinje cells. These granule cells, their axons, they form the parallel fibers in the molecular cell layer. In the molecular layer, the axons of granule cells, they divide in two and form a T-like structure. They form a T-like structures. These are known as the parallel fibers. These fibers are known as parallel fibers. Parallel fibers are nothing but the axons of the granule cells. These parallel fibers, once these granule cells, they receive the impulse via Mosses fibers, the parallel fibers, they excite the three inhibitory interneurons, that is stellate neuron, basket cell, and the Golgi cell, and one Purkinje cells. The Purkinje cells, they also receive the input via Mosses fiber, from the Mosses fiber. The output of this Purkinje cell is always inhibitory. The output of the Purkinje cell is always inhibitory. Now, the function of this stellate and the basket cell is to cause the lateral inhibition. These are, as I had told you, that stellate cells and the basket cells, they are inhibitory interneurons which are present in molecular layer. These, whenever they are excited, or whenever they receive the excitatory impulse from the granule cells, what they will do? They will inhibit, they will cause the inhibition, they will cause the lateral inhibition. That is, they inhibit the Purkinje cells on the sides. And this granule cells, they also send the Golgi cells. Golgi cell, they excite the Golgi cell also, and the Golgi cells, as I had told you, that they are responsible for feedback inhibition. Whereas these stellate cells and the basket cells, they are responsible for their inhibition is known. The inhibition, lateral inhibition caused by stellate cells and the basket cells is known as feed-forward inhibition. It is feed-forward inhibition. Why we say it's a feed-forward inhibition? Because after excitation of these basket cells and stellate cells, they cause the neurotransmitter released from these cells, which is responsible. It causes the lateral inhibition and increases these cells, they inhibit the Purkinje cells, which are on the sides of the main Purkinje cell, from which the inhibitory impulse will be conveyed to the dentate nucleus. Now, these Purkinje cells, they are receiving two output. One is mossy fibers, as I told you, and the climbing fibers. Now, mosses fiber, it is receiving the excitation via, from the mosses fiber, they excite the granule cells, and granule cells, they excite the Purkinje cells. Once via these parallel fibers. And the climbing fibers, they straight away, they synapse with the cell body and the dendrites of the Purkinje cells. Whereas these mossy fibers, first they will excite the granule cells, and then the parallel fibers, they synapse with the dendrites of the Purkinje cells. And these, from here you can see the lateral inhibition of the basket cells. That is, the basket cells, they are inhibiting the adjoining cell area, and then the granule cells, they are synapsing via parallel fibers to the Purkinje cells. And the output of the Purkinje cells is through axon to the deep nucleus, that is majority of it is to the dentate nucleus. And from the output of, always remember the output of Purkinje cell, it is always inhibitory. It is always inhibitory. The Purkinje cell output is always inhibitory. And the output of dentate nucleus is always excitatory. The output of this Purkinje cells, it is always inhibitory, and the output of dentate nucleus, it is always excitatory. So these Purkinje cells, they are receiving the climbing fibers and the mosses fiber. Climbing fibers, they are coming from inferior olivary nucleus. And the mosses fibers, they are coming from they are the second order neurons from rest of the tracts. The rest of the incoming tracts, which are, they are consist of mosses fibers. The mosses fibers, in the mosses fibers, the simple spikes are generated, whereas in the climbing fibers, the complex spikes are generated. They are the long duration, prolonged action potentials which are generated in the climbing fibers. Or they are also known as complex spikes or the prolonged action potentials. In case of mosses fiber, simple spikes or the short action potentials which are generated in mosses fibers. They are weaker in nature, whereas the action potentials which are generated in the climbing fibers, they are stronger in nature. They are very strong in natures. I hope everything, this internal architecture of cerebellar cortex, it is clear to you. Let's revise it again. That it consists of three layers: the cerebellar cortex, molecular, Purkinje, and the granular cell layer. The types of cells, there are total 5 types of cells which are present in the cerebellar cortex. One is, there are 3 inhibitory interneurons, stellate cells, basket cells, and the Golgi cells. The stellate cells and the basket cells, they are present in the molecular layer and they are responsible for lateral inhibition, that is also known as the feed-forward inhibition. Lateral inhibition of the Purkinje cells, or that is also known as feed-forward inhibition. Then this Purkinje cell layer, it consists of Purkinje cells. They are big in size and they get the, they receives the input via, they are excited by the granule cells. The granular cells, it consists of two types of cell. One is granule cells, another are Golgi cells. The Golgi cells, they are excited by the granule cells and they are inhibitory, as they are inhibitory interneurons, these Golgi cells, they inhibit the granule cells and they control the impulse which are generated in the granule cells. And that type of inhibition is known as feedback inhibition. So, these Golgi cells are another type. So, three interneurons are there. Granule cells, they are excitatory in nature. Their output is excitatory, whereas the Purkinje cells, they are inhibitory. Their output is inhibitory. Purkinje cells' output is always inhibitory output. Okay. Now, the climbing fibers, the function of this climbing fiber is, what is the advantage of these complex spikes or the strong prolonged action potentials? These climbing fibers, they are responsible for learning the motor activity, repetitive, the type of activity, repetitive type of motor activity. Like when you are juggling the balls. If you are juggling the balls with 3 balls, if you start juggling in your hands, or if you are skipping, when you are skipping, in that, to learn the repetitive motor activity, these climbing fibers are responsible. They help in learning the motor activity. How they help? These climbing fibers, when we suppose we are learning the skipping. When you are learning, initially what will happen? The rope you will not be skipped properly, the rope, and the rope will wind around your legs. Now, that is, you have committed some mistake. You're jumping, the timing of going the rope down your leg and jumping, it is mismatched. Now, what will happen? The spino-olivary tract from the peripheral part, that is from the lower part, it will convey to the inferior olivary nucleus that there is some error in the motor activity. And these climbing fibers, these climbing fibers, what they will do, they will correct the rate of firing and the generation of spike potentials. The rate of generation of complex spikes, it will change. And these fibers, they fire strongly. These climbing fibers, they fire strongly because you have done a mistake while skipping the rope. And once these climbing fibers, they send this they excite the Purkinje cells and their output is also increased. The inhibitory output of Purkinje cells, it increases. As the climbing fibers, the rate of firing of climbing fibers is increased, the inhibitory output of Purkinje cells also increases, and that helps in learning the repetitive motor movements. And that corrects the repetitive motor movements. That is the role played by the climbing fibers in learning the repetitive motor movements. Next. Here you can see in this diagram that, as I had told you, that these Purkinje cells, they receive, Purkinje cell, their output goes to the deep nuclei. The deep nuclei, they receive the input. These deep nuclei, they not only receive the input from the Purkinje cells, they also receive these Mosses fibers and the climbing fibers directly excite the deep nuclei. So the deep nuclei, they are receiving the input, three types of inputs. One is directly from the climbing fibers, that is excitatory in nature, and the Mosses fiber, which is again excitatory in nature, and the input from the Purkinje cells, which is inhibitory in nature. In this diagram, you can see the effect of all these cells. That is the stellate cell, basket cell. It is inhibitory to Purkinje cells, and whereas these granule cells, the via parallel fibers, they are exciting. The granule cells, they are exciting four cells. One is stellate cells, basket cells, granule cells, and the Purkinje cells. That is, their output of granule cells is excitatory. This plus sign denotes the excitation of these cells. And then the output of the granule cells, this Golgi cells, it is inhibitory to granule cells. In this diagram, you can see that this Golgi, it is a minus sign denotes the inhibitory output, inhibitory to granule cells. That is, this is known as feedback inhibition. And these stellate cells and the basket cells, they are inhibitory to Purkinje cells, and this type of inhibition is known as feed-forward inhibition. So, the deep nuclei, they are receiving output from three output, two excitatory and one inhibitory. Excitatory from climbing fibers and the Mosses fibers. And they also receive some other inputs which are excitatory, that is monoaminergic neurons, then serotonergic neurons, and some other neurons, they also send their excitatory inputs to deep nuclei. And inhibitory input to deep nuclei is only one, that is via Purkinje cells. Next. Now, from this diagram, you can see that from the cerebellar, various parts of divisions of cerebellum, how this input, it is how this input pathways from the cerebellum, they are combining, and they are output. This is the output of the cerebellum, how this output it is governed. Now, in this diagram, you can see that the paravermal zone and the vermis, that is the spinocerebellar region. From the spinocerebellar region, the output via these Purkinje cells of these regions, that is the spinocerebellar region, they send the impulses to the interpositus nucleus and the fastigious nuclei. These nuclei, they are the deep cerebellar nuclei. The spinocerebellar region, it sends via the output of these two regions, it goes to the interpositus nucleus and the fastigious nucleus. From the interpositus nucleus, the red nucleus receives the impulse, and this vestibular area and the reticular nuclei, and the reticular, whereas this reticular nuclei, it receives from the vermis area, whereas this vestibular nuclei, it receives from both paravermal as well as from the vermis area. It receives the impulses from both the areas, vermis as well as the paravermal area, whereas this paravermal area, it sends impulses to the red nucleus also. Now, these impulses, then they, from the red nucleus, the fibers, these impulses, they are conveyed to the various descending, extrapyramidal descending tracts. That is reticulospinal tract, vestibulospinal tract, rubrospinal tract, and the pyramidal. These extrapyramidal tracts, whereas the output from the cerebrocerebellum, these Purkinje cells, they convey the output of cerebrocerebellum to the dentate nucleus. Then this dentate nucleus, it conveys the impulses via thalamus to the cerebral cortex, and it controls the activity of corticospinal tract, then corticobulbar tracts. That is the pyramidal tracts. It controls the activity of the pyramidal. That is the cerebrocerebellum. It controls the activity of the pyramidal tracts, and via both pyramidal as well as the extrapyramidal, with the help of corticospinal tracts and the corticobulbar tracts, it influences the activity of impulse carry impulses. It controls the activity of all types of descending tracts, that is pyramidal tracts as well as the extrapyramidal tracts. All the tracts, they are controlled with the help of this cerebrocerebellum. The vestibulocerebellum, the impulses, they are not conveyed to any of the deep nuclei, cerebellar nuclei. Rather, they are conveyed directly to the vestibular nuclei through via cerebellovestibular tract. They are conveyed directly to the vestibuli. Without the impulses, they do not pass through the deep cerebellar nuclei from the vestibulocerebellum. Whereas from the spinocerebellum and the cerebrocerebellum, the impulses, whatever output is generated in these two areas, cerebellar divisions, via Purkinje fibers, it is conveyed to the deep cerebellar nuclei, and from the deep cerebellar nuclei, the impulse, then they control the output of the descending tracts, and then they convey to the various areas of the brainstem as well as the cerebral cortical area, and they control the functioning impulses of the descending tracts. Whereas in the vestibulocerebellum, the output of the vestibulocerebellum, that is the flocculonodular area, it doesn't go to the output of that, it doesn't convey to the deep cerebellar nuclei. Rather, they are conveyed directly to the vestibular tract, vestibulospinal tract, and they control the activity of the vestibulospinal tract. Next. Yes. So, this is about the internal structure of and the output and the input from the cerebellum. Today we have discussed I have not touched this anatomy in detail, but we have discussed the internal structure and the divisions which are related to the physiological importance. Then physiological division or the functional divisions of cerebellum we have discussed. Then internal architecture of the cerebellum we have discussed. And then the output, the input tracks, how the input is received to the cerebellum and how the output is generated from the cerebellum, and it is conveyed to the various parts of the brain. In the next class, we will discuss the cerebellar functions and the disorders of the cerebellum. The internal architecture For understanding the functions of the cerebellum, this internal architecture of cerebellum is very important, and you must understand first that this internal architecture, because these internal architecture, they will help in the generation of the functions of cerebellum, which we will discuss in detail, the functions and the disorders of cerebellum in the next lecture. Okay, thank you very much.